Continue the complex lipid topic. I have given you assignment about function of phospholipid, but hope that your assignment will be not just a copy of these slides. Function of phospholipid as structural phospholipid participate in the lipoprotein complexes. We will discuss lipoprotein also in this lecture, which are thought to constitute the matrix of cell wall and membrane, the myelin sheet, and of such structure as mitochondria and microsomes. In this role, they impart certain physical characteristics, unexpectedly high permeability towards certain nonpolar hydrophobic molecules, lysis by surface active agents, detergent, bile salts, etc. Some phospholipids have role as an enzyme. There are certain enzymes that need tightly bound phospholipid for their action, e.g. mitochondrial enzyme system involved in oxidative phosphorylation, role in blood coagulation, uh, which type of phospholipid are important for blood coagulation. Phospholipid play an important role in the blood coagulation process. Cephalin, good. Next, role in lipid. mm, Role in lipid absorption from an intestine. During emulsification of lipid water mixture, lecithin lowers the surface tension of water, which helps in emulsification process. Thus, a prerequisite in digestion and absorption of lipid from the GIT. Role in transport of lipids from intestine. Exogenous triglyceride is carried as lipoprotein complexes, chylomicrons, and lipoprotein plays an active part in this process. Protein lipase. Role in transport of lipid from liver. Endogenous triglycerides are carried from liver to various tissues as lipoprotein complexes. Lipotropic action of lecithin. Choline acting as a lipotropic. These are various functions of phospholipids. Membrane phospholipid is source of arachidonic acid. Insulation of cofactor. These are various functions. Now, clinical importance of phospholipid, which is diphenylmethyl is already you know that it is a component of surfactant which lower the surface tension in lung alveoli. Surface activity is a phenomena by which the surface tension of the air alveolar lining interferes is lowered with expiration due to presence of DPL. At the end of expiration, when DPL is present, it prevents the alveoli from collapsing. And if DPL is absent, the alveoli radius becomes smaller with expiration and it could get collapsed. Absence of DPL in premature fetus produce collapse of lung, which produce respiratory distress syndrome. Due to deficiency of diphenylmethyl just remember this one. Now, there are ratios which help in us in describing that either the lung maturity is okay or not. That ratio is lecithin into sphingomyelin ratio. Lecithin to sphingomyelin ratio and amniotic fluid have been used for the evaluation of fetal lung maturity. If someone have a previous history of respiratory distress syndrome in their babies, so you are going to check the new pregnancy, the new baby, either that his lung is mature or not. You will check this ratio, lecithin to sphingomyelin ratio, and the amniotic fluid. This test is available. If LS ratio is greater than 2 or greater than 5, this remember this word. If it is greater than 2 or 5, it indicates fetal lung maturity. You know that lecithin preparation, lecithin synthesis occurs beyond 34 weeks of gestation in fetal lung. Before 34 weeks, lecithin preparation and synthesis is not that much to cause the fetal lung mature. So, if this ratio you get, it indicates fetal lung maturity and suggests that respiratory distress after delivery is not likely to be developed. Delivery of premature low birth weight fetus with lecithin to sphingomyelin ratio 1 below 1 or less than 1 or less than 1 indicate that the infant will probably develop respiratory distress or hyaline membrane disease. Now, glycolipid, cerebrosides, glycospengosides, these are three names. Cerebrosides occur in large amount in the white matter of brain and in myelin sheets of nerve. They are not found in embryonic brain but develop as medulation process. In smaller amount, they appear to be very widely distributed in animals. A cerebroside is consist of the following we have discussed in the last lecture, that it is composed of sphingosine, which is fatty acid of high molecular weight, and one other unit which is galactose, and sometime maybe the other unit is glucose. There is no glycerol in cerebrosides, no phosphoric acid, and no nitrogenous base. So by hydrolysis of cerebroside, you can yield a sugar, which is either glucose or galactose, a high molecular weight fatty acid, alcohol, sphingosine, or dihydrospengosine. Clinical importance of cerebroside is that it causes its metabolic defect can lead to Gaucher disease. Now, what is Gaucher disease? Gaucher disease is actually the accumulation of various cerebrosides in our body tissues. Which is Gaucher disease is typically a lipid storage disease in which there is glucocerebroside accumulation in various tissue in our body, especially in macrophage monocyte system tissue, which is spleen, liver, lymph nodes, and bone marrow. So its accumulation here cause various clinical manifestation. Gaucher disease in infancy and childhood, this Gaucher disease have further three types. Gaucher disease 1, 2, and 3. But you have to remember just an enzyme in the next slide, and that what is Gaucher disease? It is lipid storage disease. Don't remember these things. As glucocerebrosidase accumulation occur in the monocyte macrophage system, which is spleen, so the spleen enlargement will occur. The abnormal glucocerebroside and the high amount accumulates in the bone marrow, and it replaced the bone marrow. So a person will complain of bone pain due to replacement of marrow cell by histocytes that are loaded with lipids, which lead to progressive anemia, leukopenia, thrombocytopenia, bone marrow failure features. These are all the bone marrow failure features, aplastic anemia features, tendency to bleeding and secondary infection. Now, The enzyme deficient and Gaucher disease is, please, which enzyme is deficient? Glucocerebrosidase. And the accumulation of which substance occur? Glucocerebroside. Yes. Now, gangliosides, the highest concentration are found in gray matter of brain. Biomedical of ganglioside are mainly component of the membrane, therefore they can serve as a specific membrane binding sites for circulating hormone and thereby influence various biochemical processes in the cells. Now, we already know about Tay-Sachs disease that it is due to accumulation of GM2 gangliosidosis, GM2 gangliosides, and it due to which hormone, which enzyme deficiency? Hexoaminidase A. Accumulation of ganglioside, yes. Hexoaminidase, yes. Now, accumulation of ganglioside in brain and nervous tissue take place. It is a rare inherited disorder in infant. There is progressive development, idiocy, and blindness soon after birth. And the retina will look, the optic disc will be really cherry red spot, look like cherry red spot. There may be seizure, initiation, macrocephaly. Prognosis is bad and death usually occur. Now, sulfolipid. This is also a subgroup of phospholipid. They are sulfate ester of glycolipid. Lipid material containing sulfur are present in various tissue, and its metabolic defect is called metachromatic leukodystrophy, in which the sulfatide gets accumulated in various tissue. In metachromatic leukodystrophy, sulfolipid accumulates in various tissue. It is further two types, late infantile type and adult type. The presentation will be weakness, ataxia, hypotonia, and paralysis, difficulty in speech, optic atrophy, and in adult it can present in the form of psychiatric disorder leading to progressive dementia. Now, the lipoprotein is also an example of complex lipid. You are teased about lipoprotein or not? Yes or no? Various, no one teach about lipoprotein. So this is a big topic, and here we will briefly discuss it, but anyhow, you will get a proper lecture about lipoprotein in future. So I will discuss briefly, and I will clear your concept about lipoprotein, that what is lipoproteins. Various MCQs, a lot of MCQs come from lipoprotein, and you have a table in your book about lipoprotein um, regarding various type of lipoprotein, and MCQ come from that table always. So we will, because these slides are deficient about lipoprotein, I have just write their names here. Lipoprotein have, what are lipoprotein? Lipoprotein are fat soluble substances which can transport fat from one tissue to other tissue. There are further subtypes which are chylomicron, VLDL, LDL, and high density lipoprotein. Now, what is chylomicron? When you eat something and it contains lipids or fats. These are absorbed from your intestine in the form of chylomicron. These chylomicron made by the small intestine in the feed state. When you feed and you get lipids or fatty acids, uh, it absorbs in the form of chylomicron from your intestine, and it goes into the lymph vessel, then moves into the blood. These chylomicrons are rich in triglyceride. This is a simple schematic diagram of chylomicron. Chylomicron contain triglyceride. Either these triglyceride uh, goes for the preparation of fatty acid, or it goes to the liver for various metabolic function. And these fatty acids are either deposited in your adipose tissue, or it will give some energy to your muscle and heart, these fatty acids. So chylomicron are rich in triglyceride. Please note this point, and it helps in transporting triglyceride from intestine to liver and various tissue. Now the second one is very low density lipoprotein, which is made in the liver from excess dietary carbohydrate. Chylomicron export exogenous triglyceride into various tissue, and VLDL transport endogenous triacylglycerol triglyceride from liver to various tissue. So, which triglyceride is prepared in the liver and it is transported to various tissue in the form of VLDL. It is secreted into the bloodstream. It is also rich in the triglyceride. Various MCQs which come, I will show you later that which type of MCQ can come from chylomicrons, VLDL, and etc. Function of VLDL is to deliver triglyceride to body cells. Next, contain ApoB1. Now you will ask that what is ApoB100? This is an simple picture of lipoprotein. It have an outer core and it have an inner core. The outer core, peripheral core, is composed of phospholipid. This is phospholipid. This is the polar and this is the non-polar head. And this is the cholesterol. The outer core is composed of, this is a simple picture of lipoprotein. The outer core is composed of phospholipid and cholesterol. The inner core is composed of, this inner core is composed of triacylglycerol, cholesterol ester plus cholesterol esters, and there are proteins which are embedded either in the or which are outer lipid layer. So these are either integral proteins, which is called apoprotein, or either peripheral protein, which are just embedded or which are just attached to the outer surface of the phospholipid and cholesterol. What are lipoprotein? Lipoprotein are composed of outer lipid, outer core and inner core. Outer core is composed of phospholipid and cholesterol. The inner core is composed of triacylglycerol plus cholesterol esters. And a protein portion have either integral protein, which is called apoprotein, or peripheral protein, which is attached just to the surface of the phospholipid. So this is simple diagram. Now we will discuss VLDL again. It is composed of, it is called very low density lipoprotein. It forms in the liver, which which help in transport of excess dietary carbohydrate and protein from liver to various tissue. It is secreted into the bloodstream, which is rich in triglyceride. This apo, in case of LDL, this apoprotein is apolipoprotein B. In case of VLDL, this apoprotein is ApoB100. So remember it. Every lipoprotein have a different apoprotein, and it is named according to the type of lipoprotein. It is similar. Very low density lipoprotein is similar to chylomicron, but made by different tissues. It is made mainly by the liver, and chylomicron are prepared in the intestine. Now, VLDL are rich in triglycerides. Either it helps in formation of fatty acid, which is deposited in the adipose tissues, or either it will give energy to the heart and muscle, or either it will goes either it will goes to the liver, and glycerol will form in the liver, and it's stored, and it will help in preparation of various glycophospholipid further. Once VLDL loses much of its triglyceride, it becomes LDL. Now, low density lipoprotein made in the liver as VLDL arise from VLDL once it has lost a lot of its triglyceride. Once VLDL lost his triglyceride, it is converted to LDL, and it is also secreted into the bloodstream, which are rich in cholesterol, function deliver cholesterol to all body cells. You आपने सुना होगा कि what are bad lipoprotein and what are good lipoprotein. So LDL is bad lipoprotein, or good LDL is bad lipoprotein because it, it transport various it transport fatty acid, triglyceride, cholesterol from liver to the tissue, where it stucks and it leads to atherosclerosis. HDL is good. Yes. Now this is a simple diagram. It's already we discussed that. Now HDL, high density lipoprotein, which is also made in the liver and small intestine, secreted into the bloodstream, function pick up cholesterol from body cells to take it back to the liver, reverse cholesterol transport. It works against low density lipoprotein. So HDL is good lipoprotein and LDL is bad lipoprotein. Potential to help reverse heart disease. LDL is going to worse the heart disease, and LDL is going to make atherosclerosis and make the vessel blockage, and in response the HDL is working against the LDL. When you get dietary lipids in the intestine, chylomicron helps in transport of that dietary lipids. Emulsification of fatty acid occur in the intestine, and chylomicrons help to absorb these lipids, these digestible fatty acid in the form of chylomicrons, which have apoprotein in the form of B48. Remember this one? It goes through capillaries, and lipoprotein lipase work on chylomicron, and it converts chylomicron into chylomicron remnant, and it ultimately get into the liver, where liver is a metabolic machinery, and it converts the chylomicron remnants, which have rich in triacyl glycerol, to be converted either to carbohydrate, either to proteins, or either at help in storage of that fatty acids. Now, I will show you Harper book, that which we will just briefly look at that what we are taught. So, the lipid are heterogeneous group of compound including fat, oils, derived waxes, and related compound which are related more by their physical than by their chemical properties. Now we will see that lipids are classified as simple or complex. Simple lipid further classified into fats and waxes. Compound lipids is further classified into phospholipid, lipid containing, in addition to fatty acid and an alcohol, a phosphoric acid residue. You know it. And it is further classified into glycerophospholipid and sphingophospholipid. The other type is glycolipid. Complex lipid include glycolipid. Other complex lipid are sulfolipids, aminolipids, and lipoprotein. Precursor of derived lipids are fatty acid, glycerol, and steroids or other alcohol. Now, leave this. Now, what are unsaturated fatty acid and saturated fatty acid? These are various saturated fatty acid. HDL, pick up cholesterol. No, HDL pick up cholesterol from peripheral tissues and add to it transport cholesterol from peripheral tissue to the liver. मैं आपको सिर्फ ये बुक इसलिए दिखाना चाहता हूँ कि मतलब what we teach here we mention these are simple بس دغه خبرې دي دا کمی چې په هارپر کې دا سلایډز چې دي دا موږ د هارپر نه د دې بوک نه جوړ کړي دي کنه نو کم چې امپورټنت خبرې وي ما تاسو ته منشن کړی چې کم په دې کې د کار خبرې دي ٹھیک شو نو جست سمپل اوورلوک اسې دی له ورکو چې موږ څه ویلو نو سیچوریټډ فیټي اسید دا ټایپس دي بیا unsaturated fatty acid further classified into monounsaturated, polyunsaturated. Now these are various fatty acid which have their number and family. You can ask about family, the total omega 9 fatty acid. So remember this one. What are omega 6 fatty acid? Remember this one. Leave this now. This is the slide which we have discussed that either the X group may be choline, ethanolamine, serine, and myoinositol. Diethylmethyl is a very effective surfactant agent and is a major constituent of the surfactant preventing adherence due to a surface tension of the inner surface of the lungs. Respiratory distress syndrome, this is a clinical important point. This is plasmalogen, in which the X group is ethanolamine. We have discussed it. Deficiency of sulfolipid. Disorder of metabolism of sulfolipid leads to which disease? Metachromatic leukodystrophy. So just remember their clinical name. Sphingomyelin are found in the nervous system. Sphingomyelin are found in large quantity in the brain and nervous tissue. These are all. I will send you this book in case of any difficulty. You can read this book. These figures we have discussed in the first lecture, that what is ceramide, what is sphingomyelin basically it looks like, and what is lysoleucine. This is a structure of cerebroside, galactocerebroside. Now I will ask some points from you, and please write it, please. Which of the following lipoprotein are rich in lipid content? Number one, HDL. Number two, LDL. Number three, VLDL. And number four, chylomicron. Please write this MCQ. Which of the following lipoprotein are rich in lipid? Have more lipid contents. Number one, HDL. Yes, chylomicron. Number two MCQ is which of the following lipoprotein contain high protein contents? Which of the following lipoprotein contain high protein content? Number one, HDL. Number two, LDL. Number three, VLDL. And number four, chylomicron. It is the reverse of the first MCQ. High protein content. I'm going to show the slides again. Now, HDL have HDL have high protein content as compared to the other three, and chylomicron have high lipid content as compared to these three. Please remember it. The other MCQ which can come from lipoprotein is that every, I have already told that every type have their separate apoprotein. In case of chylomicron, this apoprotein is apoprotein 48. In case of HDL, this apoprotein is apoprotein A. In case of VLDL, this apoprotein is B100. In case of LDL, this apoprotein is apoprotein E. So remember the separate apoprotein names for each of the lipoprotein. Now, and which is good lipoprotein? HDL. And what is bad lipoprotein? LDL. ډیر زیات کمپلیکس او ډیر زیات بور ټاپک دی. بهرحال خو تاسو به په دغه کې وګورئ خپل په بوک کې، او زه به درته د هار په نوم درولېږم که څه دغه پکې. ApoB100 is apoprotein of VLDL. And apoprotein A is protein of HDL. Chylomicron have apoprotein B48. Apoprotein B48. This apoprotein is different for each lipoprotein. In case of LDL, this apoprotein is B100, apoprotein B100. In case of HDL, this apoprotein is ApoA. And some other type also have, but the main type is ApoA. LDL have apoprotein B100.